So this is today's topic, and what I really would like to do today is to describe a little bit different variants of modeling and simulation techniques that can be used to better predict or better understand solubility, permeability, and how you potentially can make use of these to also understand formulation pathways. And I'd like to start with acknowledging the group in Uppsala, of course, because a lot of what I will talk about today is our own work. I will, of course, provide references to other sources as well, but these people here are part of my team. Not everyone was present. This feels like ages ago. It's actually only like 8 months ago, but we had this group day, which today seems to be completely unrealistic to have again for the coming year or so. So I will present different kind of methods, as said, and focus on the computational part, but they heavily rely on the high quality experimental data. So this group contains very skilled experimental scientists as well as computational scientists who collaborate strongly together. I also would like to acknowledge the people that we have collaborated with. And today, when I talk about formulation pathways, I will focus on lipid-based formulations. The things that we have done with lipid-based formulations, very similar things we have done with amorphous materials and amorphous solid dispersions. But I didn't have time to squeeze it in for the 40-minute presentation that I will give. So if you're interested in hearing more about amorphous formulations, please let me know and I could provide information to you about that. But with regard to the lipid-based formulations, I have a strong collaboration with the people at Monash University. So I would really like to thank Chris, Ben, and Bill at Monash for that good collaboration. And I will also today describe a little bit about different kind of solubility predictions in intestinal fluids. And for that, we have a long-standing collaboration with Patrick Augustins at KU Leuven in Belgium. And of course, we are very happy for all of the funding that we get, and particularly for this platform, the Nordforsk funding has been essential, and the Vinnova funding for the Swedish Deliver. So with that, I will start to just describe a little bit, and I will try to make this from very basic and making it a little bit more advanced as we move along. But as said, this is an introductory seminar for the topic. So just a little bit of an explanation around the solubility issue and molecular properties around solubility, because that's really where we come from, the solubility side, and that is what has led us into the formulation pathway. And if we look into the reasons for poor solubility, one can divide them into two main categories. One of them being that the solid state is the limiting factor, and the other one being that it's a solvation limited property or limitation for the solubility. So if we look into the solid state limited, that's really about the problem for the compound to dissociate from its crystalline material. And the property that we typically make use of to reflect this is the melting point. So we know that compounds that have a melting point that is above 200 degrees Celsius, they have a strong influence of the solid state on the resulting solubility. So they are called solid state limited, and sometimes more commonly used or popularly scientifically described as break dust molecules. The ones that are highly lipophilic, and they are typically the ones that we will then move forward for a lipid-based formulations, they suffer from a solvation limitation in their solubility. So it's really about the poor interactions between the water phase and the compound as such. So here we make use of the log P as a determinant to identify if we have a solvation limitation. And typically we make use of a cutoff of log P of 3 or log D of 3 if we are investigating or exploring the pH-dependent solubility. So compounds that have a log P greater than 3, they are solvation limited, and sometimes they are called greaseball molecules in the literature. And unfortunately, of course, we have those that have both these limitations, and they are really very difficult to formulate and pass through in the development chain. And typical such compounds are highly hydrophobic and largely aromatic compounds. So the melting point and lipophilicity influence on solubility is very well captured by the general solubility equation that we have here in the corner. which relates the intrinsic solubility. So this is now the solubility of the neutral species to the melting point and the lipophilicity. And this was established by Jane and Jalkowski about 20 years ago by now. So this is for pure water system. But in real life, we are typically interested in solubility in intestinal fluids. And now it becomes much more complex. So in the intestinal fluid, we have a lot of different colloidal structures or non-aggregates that are composed of phospholipids and bile salts, cholesterol, as a result of a bile secretion. And so these forms different kind of colloidal structures, anything from vesicular to micellar structures. And these can then solubilize the drug. So if it's a highly lipophilic drug, we know that it will be keen to interact with these colloidal structures, and that can potentially increase the solubility then in the intestinal fluid. This is a very dynamic system, so the solvation capacity throughout the intestinal tract is not the same. So we both have the pH gradient going from low in the stomach to relatively high in the colon, but then also these differences in the bile composition and also absorption of both products like the bile salts, but also of water. So it is a changing system which will then change the solvation of capacity of the solvent. This will be even more complicated if we eat or if we take a formulation which contains a lipid. So if we have food intake or a lipid-based formulation intake, that may trigger both the bile secretion, so we have more of the bile components coming out, but it will also start to become digested by enzymes that are released by the bile. These products that are formed, the digestion products, they will then also potentially be incorporated into these micellar structures and further impact on the solubility. So in real life, this is a very complicated system already in the fasted state, but in the fed state or when exposed to particular formulations, it may be even more complex. So then a question that arises, of course, if it's possible to predict intestinal solubility, so solubility in the different regions of the intestine. So I will start with go through a number of different techniques that you can make use of, or I will start actually with going through the multivariate data analysis technique, and then move on to showcase how we can make use of MD simulations or molecular dynamics simulations. I will not go into super detail into each of these. I just would like to give you a feel for what the workflow is and what you can do. And then if you're interested in having more details, we can, as said, provide new webinars that go into details in each of these methodologies. So the multivariate data analysis approach has been used for quite some time to predict solubility in water-based systems, permeability or maybe transporter interactions. And it really stems from the chemistry field where this kind of approach has been used for decades to predict activity. So what is typically referred to as quantitative structure activity relationships, QSAR. So when we use the same kind of approaches, we talk about QSPR, where the P is property, so property relationship. And property, as said, can be anything, solubility, enzymatic patterns, or it can be permeability. So just to take you through the workflow when you develop these kind of models, it's like, of course, that you first have to decide on what kind of data set you're interested in exploring. And what we always do in our group is that we focus on drug-like compounds. So that is our highest priority. If you would work in another field, it may be other kind of data sets that are more important. We have also focused on developing selecting as diverse compounds as possible to expand the chemical space as much as possible. If you're sitting in a pharmaceutical company, maybe it's more interesting to actually have dedicated chemical spaces dependent on which target you are exploring, because by doing that, maybe you can increase your accuracy in the prediction. So this is a little bit dependent on your question and the application of your model, which kind of diversity and space you'd like to cover. So when you have decided that with regard to your data set, then of course it's about generating the experimental data. And this is absolutely the most time-consuming part of the whole method development. So it's very crucial that we have high quality of the experimental data, because the models that we get in the end will never be better than the experimental data. So it's highly recommended to make use of the same standard procedure when establishing the response parameters, so to speak. So measuring solubility, for instance, in the same way, or the permeability in the same lab, because there will be differences when one starts to compile literature sources and to build up the data set. So it's preferred that we actually have made high quality determinations ourselves, or take high quality data from another lab. Then we do calculations. Typically here we make use of different kind of molecular descriptors. That most commonly used are the 2-dimensional and 3-dimensional descriptors. This is a very quick step in the whole process. And when we have these two data, then we combine them with a statistical method to try to relate then the molecular properties to the response parameter experimental data. This can be done by very many different approaches, so different linear approaches or non-linear approaches. And I would say that actually the deep learning approach is coming also within this field. So there are a few publications coming out now, also making use of machine learning techniques as the deep learning method. This demands a high number of experimental data points. So that is typically what has been restricting the field, especially when we talk about so those properties are solubility and permeability. So you develop your model, and you do that by dividing all your experimental data into two sets. So you divide the experimental data into a training set, and this training set is used to develop the model to find those relationships. And about 1/3 typically we keep for a test, and that's then just called the test set, and that we use to do a validation. So if these test compounds now, which have never been seen by the model, so to speak, if they are well predicted by our model, yeah, then we can approve the model and start to use it for new unknowns. So that's the workflow around those multivariate data analysis techniques. So we made use of, in this case, it was a PLS model to explore around if it's possible to predict solubility in those more complex fluids, such as the human intestinal fluid. This was a few years back. It was in collaboration with AstraZeneca, and AstraZeneca had measured a quite significant data set for their solubility in aspirated intestinal fluid. And what they have done is to aspirate from the small intestine, then take the aspirates from several different volunteers, pull them into a beaker so you get a larger volume, and that is now your solvent for your solubility test. As you can see here on the screen, we have two different colors. So we have light blue here for the training set. So if we go back, the light blue ones are the one that was used in this step to develop the model. And then the dark blue ones are the ones that we have used for validation. So that's our test set. And as you can see, we got a pretty good model here based on 9 calculated descriptors. So this is just making use of SMILE strings as input to calculate a 2-dimensional or 3-dimensional descriptor. So of course it's good to get models where you can actually predict the solubility and get an estimate, at least for your compound, what the solubility in the small intestine would be. But it's also other things that you can get out of this model, these kind of models, that may be more interesting at times for academicians than the absolute value. So that is, so what are these different properties that are driving solubility in these fluids, or maybe limiting the solubility in these fluids? So when we made that kind of analysis, I mean, it was quite logical things that came through for driving solubility. We could see that it was the number of intermolecular hydrogen bonds. So it's simply how easy it is for this compound to interact with the water phase in this solvent. And then it's the number of nitrogens and oxygens, and you can view them as anchor points for the water during the solubilization of the compound. But it was also noted that it was important for a compound to be well dissolved in the solvent, that these anchor points were spread out over the molecule, so that sort of the water can grip different parts of the molecule and hydrate that one. When it comes to limiting solubility, we could identify that size and branching is a limiting factor. This has to do with the cavity that the solvent has to open up to incorporate this molecule. But also aromaticity was found as important, and it's quite clearly visualized here in this figure that if you have these high aromatic surface areas or like flat rigid surface areas, they cannot really shield or hide from the water. They have to sit there with all these hydrophobic atoms clearly exposed to the water, and of course it's a huge energy penalty for that. But it also informs about some other things, and we know that these kind of structures are quite good for forming a strong crystal lattice. So to some extent, we may also have the result of aromaticity to pointing towards the difficulty already to let go of the crystalline state, to dissociate from its crystalline lattice. So this kind of information one can also get out from the models that are developed based on multivariate data analysis approaches. It's not only the quantitative predictive value, it's also about understanding why a compound ends up down here as a poorly soluble compound and why another one is highly soluble. Okay, so we then wanted to look into if we could combine these kind of relatively advanced computational models for prediction of intestinal-like solubility together with permeability. And this we did for relatively modern compounds. So the data set that we selected here was taken or extracted from the patent literature to make sure that we had relatively contemporary compounds and not relying back on more old marketed compounds. So this was based on a literature search, and what we did was to predict solubility now in the fasted state simulated intestinal fluid. So this is a made up solvent that is mimicking the small intestinal fasted state, and predict permeability. And in this case, we used a model that was predicting the permeability, human permeability. These two models that were used are sitting in the Dept predictor software that is provided by Simulations Plus. So this is not our own models. These are commercially available models. And again, it's only the chemical structure that is needed of the compound to predict the values. We decided to go for analyzing this in kind of a BCS or biopharmaceutical classification manner, but we wanted it to be a little bit more discovery-like. So what we did here was to include also intermediate classes in the system. And if you're interested in the details, you can go to this paper and see the cutoff limits that we have used here. But just to have a quick overview, one can say that compounds that are up here in the green corner would be BCS1-like. So they would be predicted to have high solubility and high permeability. Whereas down in this corner, we are in the BCS2-like system. So here it's BCS3 and down here 4. And if we looked into all these 1,600 plus compounds, we could see a pattern that is more or less the same as what we would expect from analyzing marketed drugs, that about 40% are up in the BCS1 corner, that 40% are down in the BCS2, and then we have a division between 3 and 4. But if we instead look into this in a target specific manner, a completely different pattern shows up. So this is the graph that I showed you in the previous slide. Now we have divided into different targets or different ligand spaces, one can say. So here we are looking into the 5-HT2C ligand. So this is serotonin. Like more or less all of these were predicted by these computational models to be BCS1-like. If we look into cholesterol, esterol, transferryl protease inhibitors, more or less all of them were predicted to be BCS2-like, and a set of macrolides were predicted to be BCS3-like. And of course, as said, this was all patented literature, so we don't really know the experimental data, so it's difficult to really prove that the predictions were right. but most of us know that macrolides, antibiotic compounds, are typically viewed as being highly soluble but poorly permeable. So at least this one sends a signal that it's quite good predictions. So then there's a question if there's anything else one can extract from these kind of models. And in this ADMET predictor, there is also a model for prediction of the fed state simulated intestinal fluid. So what we then did was to analyze to what extent these classes would change if we instead would have the fed state solubility data applied. So this is just one example of a BCS2 then that had more or less all of its compounds sitting in the BCS2 in the fasted state, where almost all of them went up to a BCS1 in the fed states, showing that those lipids are crucial for solubility of these compounds or these compounds. So in the case of going from fasted to fed state, what one is doing here in the medium is to increase the bile salt and the phospholipid level. So that's in principle the main difference. And there's also a smaller difference in the pH between these two. So we were then interested in seeing if this could be a signal for a formulation pathway moving forward. So for instance, if we see that a compound has this strong effect going from fasted to fed, would these kind of compounds also significantly benefit from being formulated as lipid-based formulations? And as said, we don't have the experimental data. So what we did was to do a retrospective study where we looked into similar compounds or analogs or ligands to these different targets and see how they were developed and formulated. And indeed, what we could see was that those serotonin-like compounds were where we predicted them to be BCS1-like. Also, there are several of those on the market that are formulated as conventional tablets. So it's not really any advanced formulation strategy that has been necessary here to develop an oral dosage form. For two other examples that we had here, where we showed a significant improvement in the fed state and moving at least parts of some of the molecules up to the BCS1-like space, there we could also see that in different kind of preclinical studies, they had used excipients that are commonly associated with those of lipid-based formulations. So it seemed like these very simplified prediction models that we put together actually also can help us to understand potential food effects and maybe also potential formulation strategies. And as said, at least then for our industrial partners, these are commercially available modules. So it's relatively easy to transfer this to your own setting. Okay, so I will now switch and talk a little bit about how we make use of molecular dynamics to study solubilization in intestinal fluids and what we can learn from that. I will again not go into very much details because we will have several webinars on MD simulations coming up. The first one is already tomorrow. But this is a completely different, of course, modeling technique. And when we look into the QSPR models, they are very static. So you are just describing your compound with absolute numbers. Lots of different kind of fingerprints can be used, and they can be combined then through this statistical method. But the compound is described in one way only. When we look into MD simulation, of course, again, the compound is described in one way only, but we also have the dynamic setting of that compound and the dynamics, how that compound may interact with other compounds through potential non-bonded interactions, for instance. So when we do the MD simulations, we are not considering all type of interactions. That's very costly to do those calculations. So we have rationalized it, and we are looking into bond vibration, angle vibration, and torsion potentials. And then, as said, also van der Waals interactions and electrostatics as the ones that are the non-bonded. So we have made use of this to understand better the inter-individual variability in human intestinal fluid. So when I talked about the QSPR technique for prediction of human intestinal solubility, that was made based on pooled volumes or samples, as said. In this study, what we had, and this was in collaboration with Patrick Augustins in Leuven, we have the exact composition from each individual, both the time resolution of each individual, but then also the possibility to compare between individuals. So indeed, what we have done now is to select 5 healthy volunteers. So in the papers by Augustine's group, they have 20 healthy volunteers that they are reporting. And they have aspirated the duodenal fluid in this case. So it's the uppermost segment of the small intestine. And so what they do is that they first take samples from those healthy volunteers that probably looks very much like this person. So young students or maybe PhD students in Leuven. And they take the samples first for a time period in the fasted state. And then they give a meal. In this case, it's Ensure Plus. So it's a liquid meal, a nutrition drink. And after that, they continue to sample for an additional time frame. So for the same individual, you first have the fasted data and then the fed state data. So you can compare both intra and inter-individually. And as you can see, it's a huge spread, both in pH, and I think this is quite interesting to point out. Sometimes we neglect that a bit, but there's actually quite a few that have relatively low pH in this region, as well as high pH. So it's not that definite, the pH range. But also a huge spread in the bile salt and the phospholipid and the cholesterol. What we also can see is, of course, that this changes a lot, especially with the bile components and the lipid components when we go into the fed state. So much higher value of all these components in the fed state. So what we did was to pick 5 individuals based on those 20 individuals, and they were picked so that to cover as much of the individual space, again, to reflect as much as possible of this population. And we plugged this into MD simulation boxes. And in this case, we made use of what is called a coarse-grained methodology for those of you who are interested in MD simulation. But that's a way to increase the speed or increase the size of the box that we are exploring. So what you see here is actually 5 different virtual intestinal fluids mimicking exact composition of real-life fluids. And it's quite clearly visible that these differ significantly. So we go from colloidal structures here that are very green, that means that they are lipid-rich, to colloidal structures that have lots of the gray in them, and that means that they also have lots of bile salts in them. What we can see is that we have both a number of different, the number of micellar structures are very different between the boxes, but you can also see that the free form of those bile salts are very different between different individuals. And that's of course will then impact on the solubilization capacity of the particular fluid and could potentially be used to study inter-individual variability in the dissolution or maybe in the solubility profile of compounds. So if we just have a little bit of a close-up for this, we can see how it's constructed. So this is just one micelle then as an example where we can see that the lipid is forming a lipid-rich core. Inside here we have the cholesterol sitting as well. And on the outside we have more or less like a bile salt shell. And if you view this in the dynamic as a movie, you would see that those bile salts are coming and going as shuttles between those micelles. So they are hanging there relatively loose. And what we are doing with these kind of virtual intestinal fluid now is, one is, as I said, the solubilization calculation and to see what kind of variability can we expect in a population when it comes to solubility. But we are also making more mechanistic studies of looking into where does the compound sit in this particular micellar structure and what is the energy barrier to pass through and come out to the water phase. And I'm sure that you will hear more about this in the upcoming webinars. But so the MD simulations can be good to use as an add-on to those QSPRs because you get more of the mechanistic insights or molecular insights into the interaction between the API and the potential colloidal structures that are formed in the gut. So I will move over and talk about lipid-based formulations. So lipid-based formulations, the main advantage of lipid-based formulation is that it's typically used to administer your compound in a dissolved state. It can also be used to administer suspension, but that's not really the main focus of making use of this pathway. It's used for highly lipophilic compounds, and of course it is then increasing the solubilization. So it's mainly solubilization, but it may also drive supersaturation when it comes digested. So as the lipid becomes digested, that may drive release of the compound and a formation of a supersaturation that also will be beneficial for the absorption. So the way that we are currently exploring lipid-based drug delivery system, or if that is a pathway through, is to make a large number of different solubility experiments in different mixtures, and then identify through those experimental screens potential good formulation strategies. And so we were interested in seeing if this could be transformed more to a computational predictive science, to also better understand the different molecular interactions that takes place in the formulation, and switch from this experimental, both time-consuming and material-consuming approach to at least do the first screen in the computer instead. So I will start going back actually to more or less where I started with the multivariate data analysis. So as said, that has been used a lot to study solubility in water, and I showed you that we can also make use of it to study in simulated intestinal fluid or in real human intestinal fluid. And so we thought that it should be also possible to make use of similar kind of approach to better understand solubility in other solvents. So in this case, we now look into solubility in different excipients that are used in lipid-based formulations. And I should say that the ones that we have used here in the study are liquids at the temperature where we have explored them, and that's 37 degrees. So again, we did the same thing as with the flow chart I showed you with regard to how we do these kind of things. So we started with selected the data sets, and I can say that to do these kind of models, this data set is a small data set, but it's a huge experimental work to establish the 40 model compounds in 9 different excipients, and I think this took about 2.5 years to do, to get the good quality data here. And so at least that has helped us to start this work, and of course you would then see that you in the future would need to expand this data set and the chemical space that has been explored, but this is a starting point. So what we did is the same as for solubility. You calculate molecular descriptor, plug it into your computer, make use of multivariate data analysis. Again, for this one, we used the PLS as our regression model, and we did separate models for all different excipients. So the different glycerides that we were exploring had different models and so forth. So what we have learned from literature is that it's actually possible to calculate the loading in lipid-based formulation based on the knowledge of the solubility of your compound in each of the included excipients. So when we started this project, there were some sparse information in the literature about that may hold. So indeed, you can calculate the solubility or the loading capacity in your final lipid-based formulation that may be produced or developed based on a number of different excipients, based on the knowledge of the solubility in each excipient, so the solubility of the excipient 1, and the weight fraction of this excipient in the final lipid-based formulation. So it is simply an additive exercise where you can then, in the end, calculate the solubility of your compound in your formulation. So this is what it looks like. These are our 9 model compounds in 4 different LBFs, and they are covering now quite a big range of lipid-based formulation system, going from a relative lipid-rich to a relative lipid-less formulation. And you can see that this calculation, just by measuring and summing up, that you can actually get a very good prediction of this loading capacity of a more complex formulation. And the benefit here is, of course, that you then don't need to measure solubility in all different kinds of formulations, and that you actually can calculate or predict the amount that should be possible to dissolve in a particular formulation that you would like to test. So what we then did was when we had developed all these models for the different excipients, we did the exact same thing, the exact same correlation, But instead of the measured experimental data of solubility, we now had the predicted that came out from this model. So this is going back to this graph is actually the same as the one that was here. So this is when we make use of experimental data. And this is now what this kind of PLS models put out to you if you have the lipophilic formulations, and in this case, more hydrophilic formulations. So these are highly surfactant rich and more of the co-solvent in these. And here we have more of the glyceride-based formulations. And if we combine these two, we can see that we actually got a surprisingly good, I would say, prediction of the solubility in these highly complex formulations. So that's again about getting a signal of how much can I dissolve in this formulation, but it can also again be used to better understand the properties of your compound and how it interacts with your formulation. So in this case, we could extract that those molecules that are lipid-loving, so lipid-loving compounds would be the ones that are highly soluble in this setting in the glycerides. They are neutral or basic compounds. They have a melting point that is below 150 Celsius. And so if we compare, when I talked about water, what we have seen there is the cutoff of 200. Here it's a little bit less, and we are not super sure about why that is. But one of our thoughts is that it may also have, with this data set that we explored and the functional groups that were included in that, because we can see that the acidic functions had higher melting point, all of them, than this 150. So it may be both the solid state and then differences in polarities that are introduced when you go for the acid versus the base. And of course, these compounds that are to be given in a lipid-based formulation should have few polar functions. And again, what we could see about the negative properties is the elongation. So this is the size factor, which we had for the water phase as well, is negative. And aromaticity is again a negative property. And I would expect that the aromaticity here is more reflecting the solid state property than actually the problem with interacting with the lipid. So this with that it cannot really shield is not as important for the compound when it comes into lipids. But it should also, be said that those hydrophobic compounds, they are definitely not always the lipid-loving compounds. They are more or less anything phobic and mostly keen on interacting with themselves. Okay, so we still were quite interested in why it's possible to actually do this with summing up the solubility from different excipients to get the loading capacity in the lipid-based formulation. And we speculated that this may be due to that the lipid formulation is not a molecular solution, so it's not really completely homogeneous, and that we may have some structures in this that are not easy to identify by experimental means. We are now looking into the dry lipid formulation. So it's not the formulation after it is dispersed into water. It's not an emulsion. It's not the lipid-based when it's dispersed into, when you take it with a glass of water. It's the dry formulation in the capsule that we would like to explore. And a potential experimental technique could be neutron scattering to look into if there is any structures here, which we have also applied for to do, which we hope that we can do in the future. But meanwhile, we have explored it with MD simulations to see what that kind of simulation, if that would identify any kind of structuring in this solution. So this is just one example of a formulation. And so I will take you through here from the left to the right. And this is now a simulation box, an MD simulation box. I would say that this is about 10 nanometers in size, so it's a cube. And when I go from the left to the right, this box is sliced. So left is in the front, and then I'm slicing it backwards towards the end of the box. And when we do this slicing, what you can see is that here you can see a lot of green dots. This is the glyceride backbone on the lipid. And as you slice it towards the end, you can see that it sort of opens up, and you can see the chains, the alkyl chains coming into the center. And when you slice even further down, you get to see the head groups again. So what this actually is a micelle that we cut through. In reality, identifying that we have some nanostructures occurring in this lipid-based formulation. Something else that we see is that the surfactant is not really sticking into this and completely mixes. It more or less forms roads in between, like threads in between. And so one thing that we speculate around this, and we have several of these, it's not only this formulation that I show here, but what we speculate here is that when it really works to do these kind of predictions is when you have some of those nanostructures. So what the API sees, or what the drug actually sees, is one component more or less at the time, and not a complete mixture of all components. And I can just say from our experience that when you go into more of the co-solvent-based systems, those predictions become a bit worse. And to me, that is also sort of signaling that the co-solvent is probably better in sort of sitting in between here and mixing with all the different excipients that is used. So that is one of our hypotheses for the time being, that those nanostructures that are formed is the reason for why we actually can do these kind of predictions. Okay, so another technique that has been used quite significantly is the COSMO-RS, and this is a recent publication from Martin Kuhn's lab in Basel. So COSMO-RS is a fragment-based QM methodology, so it actually relates or relies on a big database in the background from which you then extract those charge densities when you are building up a new compound. So when you have a new compound, you can build that based on the information from that database. So you can do that for, in this graphical abstract, what they show is an API. But they have used this now for lipid solubility. So they have done similar parameterization of their compounds for the different excipients as well. So when you make use of this kind of approach, which is a much cheaper approach than to do real QM calculations, then this is approximately the results that you would get. So here we have the calculated solubility for a medium chain triglyceride compared to the experimental value that they have measured. And so you can see that here it's some compounds are predicted very well, and some compounds are within one log unit in accuracy. This kind of method has also been used for other solubility-related studies. So amongst others by the group in Copenhagen who has looked into COSMO-RS. So that also seems to be one computational approach to make use of to better understand or predict at least solubility. Finally, now I will just show you or comment on that lipid-based formulations, what I've talked about now from a formulation perspective, was mainly directed to lipid-based formulation for the purpose of increasing solubility. And lipid-based formulations, or at least their digestion products, may also be very helpful to increase permeability. And so that is a work that Shakavat is performing currently, and he will talk more about that later on. But just as a teaser for that particular seminar, so what we do there is to make use of those virtual human intestinal fluids. We add now different excipients to those to see how they will interact. Excipient together with macromolecular drugs to see how they interact. And also then to study what happens with these permeation enhancers, which typically are free fatty acid, C8, C10, or the snack molecule, for instance. How would that interact with the lipid bilayer, and how will that impact on the passive membrane diffusion through a particular lipid bilayer? So lipid-based formulations can be used both for increasing solubility and permeability. So these different kind of modeling simulation tools can help us to predict both absolute values for different properties, but also help us to understand better about mechanisms. So with that, I'm at the end. So I hope that I informed you a little bit about how we can make use of computational tools, and also hopefully triggered some thoughts on how you potentially can make use of it for your own research questions, but also to marry them with different kind of experiments that you are performing, or analysis that maybe you can do based on computational tools that is in line with your experiments. So I did focus today on the lipid based formulations, but as said, we have done lots of the amorphous solid dispersions as well. And I know, for instance, that Alexei has had a good collaboration there with Copenhagen within the Nordic POP. So there's definitely a lot of things going on in this field as well. So with that, I think I'm at the end. I will leave this one up that I always leave up. So this is the lab of the computational formulator, which I strongly advocate that we should educate the next generation scientists so that they make use of different computational tools when they do their formulation and drug delivery design. And of course, be well trained also in experimentals or understanding of experimental data, because they go hand in hand. It's not one or the other. So with that, I thank you and open for questions.